Scientists recently found DNA evidence that points the Black Death back to one place more clearly than ever before, the Tian Shan region of Central Asia near modern Kyrgyzstan. That result changes an argument historians carried for generations, because the answer did not come from rumor, travel tales, or guesswork, and instead came from plague DNA. For a long time, scholars argued over China, the Western Steppe, the Caspian region, and wider Eurasian trade zones, so the origin stayed broad and blurry. Then modern science entered the story with bones, teeth, genomes, and dates carved on real tombstones from the years just before Europe's catastrophe began. This breakthrough is best understood in 6 parts: the old mystery, the 2 cemeteries, the DNA evidence, the plague family tree, the ecological clues, and the new history. The old mystery mattered because the Black Death was one of humanity's deadliest pandemics, yet its launch point remained surprisingly uncertain for centuries. Europe kept many records of the disaster, but good records of suffering are not the same thing as proof of where the key strain first emerged. Historians knew where the Black Death hit Europe in the mid-1340s, yet the deeper question of origin remained wide open. The breakthrough began in the Kai Valley near Lake Issyk-Kul at 2 burial sites called Karajigak and Barana. Those cemeteries had been interesting long before DNA science arrived, because excavations in the late 19th century revealed something unusual in the grave dates. Burials suddenly spiked in 1338 and 1339, several years before the Black Death devastated Europe, so scholars long suspected an epidemic had passed through the area. Some tombstone inscriptions even used the word pestilence, which sounded dramatic but still left one major problem for historians. Medieval people used epidemic words broadly, so nobody could tell from the stones alone whether pestilence meant plague in the strict bacteriological sense or some other outbreak. That left scholars with a tantalizing clue and no final proof, because the stones hinted at plague but could not prove the bacterium. The breakthrough came when researchers extracted ancient DNA from individuals buried during that mortality spike and let the lab test what the stones had suggested. 7 individuals from the 2 cemeteries were studied, and 3 from Karajigak showed convincing evidence of infection with Yersinia pestis, the plague bacterium. That first result was already huge, because the 1338 to 1339 outbreak at the site was not just some vague medieval sickness, and was real plague. 2 of those infected individuals yielded genomes strong enough for deeper evolutionary analysis, and a third lower coverage sample still supported the same diagnosis. That is why the discovery felt like a true reveal, because the inscription said pestilence and the DNA now proved those graves really held plague victims. But the second result mattered even more than the first, because scientists did not only confirm plague, and also found where that strain sat on the plague family tree. When the genomes were compared with other ancient plague genomes and with modern Yersinia pestis diversity, the Karajigak strain landed in a stunning position. It sat at, or right beside, the ancestral node that gave rise to the major lineage explosion associated with the Black Death. In simple language, the bacterium in those graves was not a distant cousin passing through Central Asia and was extremely close to the direct source line. That is why historians and genetic scientists paid such close attention, because the graves were showing a strain right next to the pandemic source. Earlier research had already revealed a puzzle sometimes called the 4-branch polytomy, a dramatic split where several major plague branches appeared. The time and place of that branching event had remained uncertain, and the new genomes helped narrow both the clock and the map. By placing the 1338 to 1339 strain right before that big branching event, the study tied the diversification to the first half of the 14th century. It also strongly linked the emergence of the Black Death lineage to Central Eurasia, rather than to much more distant and speculative locations. The dates on the graves made the result even stronger, because researchers were not guessing the century from rough soil layers and had inscriptions fixed to 1338 and 1339. Those inscriptions also linked the dead to local Christian communities connected to wider Eurasian trade networks through travel and movement. This was not a sealed mountain pocket cut off from movement and trade, and that detail matters a great deal for pandemic history. Pandemics do not emerge from microbes alone, because they emerge when microbes move through roads, markets, animals, people, and long-distance contact. The Tree Valley sat close enough to trade corridors that a local reservoir event could plausibly escape into much wider circulation. Modern plague ecology made the discovery even more convincing, because the Tian Shan area and neighboring Central Asian zones are known to hold long-standing plague foci in wild rodents. That does not mean researchers can point to one exact medieval marmot or one exact burrow, but it does mean the region fits plague ecology in a real way. The ancient DNA was not pointing to a random place with no plague background, and it was pointing to a landscape where plague already makes biological sense. The medieval genomes also showed close relationship to lineages found in the broader region today, which strengthened the case for a central Eurasian reservoir. This is where the discovery changes history and not only biology, because written sources alone had kept the origin story broad for a very long time. Ancient DNA turned that wide debate into a much narrower one, and shifted the story from one theory, among many, to the best supported source region yet identified. It also pushed the Black Death story out of a mainly European frame, because Europe recorded the catastrophe well, but the strongest source signal now points farther east. That shift matters because pandemics are continental events shaped by shared systems, and not only by the places with the loudest surviving archives. The people buried at Karajigak and Barana were not side characters in somebody else's history, because their bones and teeth became evidence for the microbial prehistory of a global disaster. Another important point is the difference between an origin region and a full travel route, because the study did not claim that one tiny cemetery alone launched the entire pandemic. What it did show is stronger and more careful. By 1338 and 1339, a plague strain extremely close to the direct ancestor of Black Death genomes was present near Issyk-Kul. That means the region was tied to the emergence event itself and not simply touched by a later wave passing through. Scientists still debate the exact first human chain of transmission, the speed of spread, and the local steps between reservoir and wider pandemic. Yet, after this study, the question of the most convincing source region became far less open than it had been for generations. The phylogenetic impact was huge too, because the branch expansion linked with the Black Death accounts for a striking share of later plague diversity. In one analysis, the descendant branches from that diversification made up roughly 40% of the overall phylogenetic diversity in the modern Yersinia pestis dataset studied. That number shows the event was not only catastrophic for humans, and was also a major turning point in the evolutionary history of the pathogen itself. In other words, the Black Death did not only kill on a vast scale, and also helped shape the later family tree of plague for centuries afterward. The science behind this breakthrough is also a story about methods working together, because no single clue would have been strong enough on its own. The tombstones showed an epidemic happened, the DNA proved it was plague, and the phylogenetic analysis showed why this particular outbreak mattered globally. The ecological background explained why the region made sense as a reservoir zone, and the trade setting explained how a local outbreak could connect to wider movement. Put together, those clues formed a chain far stronger than rumor, traveler stories, or map speculation alone in medieval Eurasia. This is why the discovery feels modern in the best sense, because science did not replace history and instead joined archaeology, inscriptions, demography, and evolutionary modeling. The old debates were not foolish, because scholars were working with incomplete tools and doing the best they could with written evidence. Once ancient pathogen DNA became available, the map of possibility changed, and the old mystery became much sharper. The result is both humbling and exciting, because a question that survived for centuries became clearer when microbes left evidence in human remains. It also shows how fragile big historical stories can be when they depend too heavily on the places with the loudest surviving texts. Europe's records once made Europe feel like the center of the whole origin narrative, but the DNA now pushes the strongest early signal into a broader Eurasian setting. That broader setting includes environment, animal reservoirs, trade corridors, migration, and communities living far from the later European centers of written memory. The Chi Valley cemeteries therefore matter on two levels at once, because they preserve local human tragedy and the earlier edge of a world-changing pandemic line. Even the timing feels chilling, because these people died in 1338 and 1339, just years before the Black Death roared into Europe in the mid-1340s. Their graves now read like a warning signal placed on the road before the largest wave reached Europe. The discovery also changes how historians can summarize the story for the public, because saying the Black Death started somewhere in Asia is now too vague. A better summary is sharper and backed by evidence. The strongest known origin signal now points to the Tian Shan region of Central Asia near present-day Kyrgyzstan. That sentence does not solve every root question, but it changes the center of gravity in the debate. It also gives the word pestilence on those old stones a new weight, because the inscription was pointing toward plague at a decisive moment. The dead from Karajigak became witnesses across 7 centuries, and their remains carried the bacterium's history longer than any written chronicle. That is one reason this finding feels so powerful, because it turns silent graves into data that rewrite a global history lesson. There is also a lesson here about pandemic memory, because people often remember the place where suffering was best recorded and forget where the chain first sharpened into catastrophe. Ancient DNA helps correct that habit by following the pathogen itself instead of only following the loudest documents about the aftermath. The Black Death will still be studied through ports, caravans, trade hubs, and later regional outbreaks because those root questions still matter. But the big origin question is now far less foggy because the best evidence no longer points everywhere at once. It points with unusual precision to a Central Asian outbreak in 1338 and 1339 that sits beside the ancestral source of the pandemic lineage. That is why this was a real breakthrough reveal, because scientists did not only find plague in old bones and found plague in the right place on the family tree. Once that family tree position became clear, the old debate changed shape immediately, and the cemetery stopped being a curiosity. That change is what modern historical science looks like, because a local burial spike, a few words on stone, and extracted DNA combined to redraw a famous map. Science did not merely add a footnote to the Black Death, and instead redrew the opening chapter for global history. Before this study, scholars had to weigh rumors against trade logic, which meant every map of origins still carried a wide zone of uncertainty. After this study, they could anchor the debate in genomes taken from named years and epidemic graves marked by inscriptions. That kind of anchor is rare in medieval disease history because date, place, pathogen, and evolutionary position do not often line up this cleanly. The find also reminds us that one cemetery can matter globally when the strain inside it sits at the hinge point of a pathogen's expansion. Karajigak was a local tragedy, but it was also a window into the moment before plague branches spread much wider. Varana helped frame the same regional setting, showing that the mortality spike belonged to a real outbreak landscape and not a single isolated grave. The words on stone, the extra burials, and the ancient DNA each mattered, but together they became far stronger than any one clue alone. That is the power of combining archaeology with genomics, because the past stops being only narrative and becomes testable evidence. It also changes classroom history, because students can now point to a specific Central Asian source region instead of repeating somewhere in Asia. The Tian Shan region near Kyrgyzstan is not a dramatic guess and is the strongest known source signal scientists currently have. That makes the discovery a map-changing result rather than a small lab curiosity for Black Death studies worldwide. The Black Death still needs route studies, port studies, and trade studies, but the starting zone is far narrower now. In that sense, the DNA did not finish every argument, but it ended the old version of the biggest one. A centuries-old mystery became a smaller and sharper question the moment the plague genome was placed beside the ancestor of the pandemic line. For centuries, the opening of the Black Death looked like a dark fog at the edge of the map. Now the fog is thinner, because a dated Central Asian outbreak can be tied directly to the lineage that later spread outward. That does not erase every missing step, but it changes the starting frame for every later route discussion. Few medieval mysteries have been narrowed so dramatically by direct pathogen evidence taken from human remains and dated graves. That is why this discovery belongs in the category of true historical breakthroughs and not minor updates in plague history. The result is not that every mystery vanished, but that the biggest mystery now has a much tighter and better supported answer. For Black Death history, that shift from wide guesswork to narrow genomic evidence is a major historical turning point. If you want the next part of the story, watch the video on how plague moved from a regional outbreak into a continental catastrophe, because the route story begins where this DNA story ends.